So, before we talk about how proteins are made, first of all we need to talk about some terminology which describes the structure and function of DNA. You can see the key terms down the left hand side. These are the things that I'd expect you to know at the end of this slide. Here we have a pictorial example of the structure of DNA. The double helix term, which is first of all here, describes this double strand structure which intertwines in a spiral shape, and it's called double helix because it's in that helix shape, but also it's got a double strand of this blue ribbon spiraling all through it. That's what we describe the structure of DNA as to be. It's a double helix. Now this blue ribbon here forms the backbone of DNA, and that's referred to as the sugar phosphate backbone. So it forms the backbone of DNA. I like the sides of a ladder. It's the rigid foundation for the molecule. The complementary base pairs are these colours here that you see that extend out from the sugar phosphate backbone. So they extend out to the sides, and they're one of four: adenine, thymine, guanine, and cytosine. So they're the four base pairs that you can get, and there's only four on DNA. Adenine always pairs with thymine, guanine always pairs with cytosine. Now, thanks to Georgia, we've got a nice way of remembering that. That's Chris Giel, awesome teacher, CG AT. Chris Giel, awesome teacher. My initials are in your DNA, don't forget that. So that's what we mean by complementary base pairs. Complementary base pairs. They complement each other, and the base pairs are always found in those matching letters. So you never, unless there's a problem, get G with T and A with C. You always CG AT, and they can be reversed on different sugar phosphates. So you could have, like the diagram shows, you can have C over here, and sorry, where are we? So that would be A and T. You can likewise have T and A on the different sides, if you see what I mean. So that's fine. Now, one part of the sugar phosphate backbone and one base is called a nucleotide. That is the building block of DNA. So if we were to, let's say, cut out, let me see, this bit of sugar phosphate backbone and this yellow base here, that would be a nucleotide. And hopefully you can see that DNA consists of a repeating unit called a nucleotide, which builds up the double strand of DNA. So that's what a nucleotide is. It's a building block of DNA, and it consists of the sugar phosphate backbone and a base. Now, the join between the two bases, so where they join here and here and here and here, is formed by what's known as a hydrogen bond. So let's say this is also the sugar phosphate backbone, and we're looking at, I don't know, any point in this double helix. So you've got sugar phosphate backbone down the left, call it blue because it's blue on the diagram, and then you've got let's say thymine here, which is green, and adenine, which is yellow. Now they're joined together in this sort of this gap here by a hydrogen bond. Now, for those of you that have had my lesson on atoms and bonding, you can just see that hydrogen just needs to gain or lose an electron to be able to join together. So if you don't understand that, it doesn't matter, but all you need to know is where that hydrogen bond is. It joins the two base pairs together. Now, let's delete some of these. It's getting a bit messy. The next key term on our list is the triplet. Now, triplet should be relatively straightforward because a triplet or triplets means 3, and a triplet of DNA means basically it's 3. I'm going to change the colour of my pen because you can't barely see it. 3 base pairs together. So they come in 3s. So the next 3 would be there. That's the next triplet on this, and there's the next 3. So a triplet is 3 base pairs following on from each other. And it's really important because a triplet is what codes eventually for an amino acid. Okay, so this triplet would code for amino acid, this triplet would code for a different amino acid, this triplet would code for amino acid. So hopefully you can see that really the function of DNA codes for our proteins, that in the way that the sequence of the base pairs in 3s codes for amino acids, and when you join those amino acids together in the right order, you get a specific shape of protein. So that's what a triplet is, and I'll be talking about more about that in the future slides. And hopefully I don't really need to go into those key terms, but adenosine, cytosine, guanine and thymine are your base pairs, and they match up in complementary style, CG 80. Okay. So before we move on to the fine details of how proteins are made, I thought it'd be useful to sort of explain the big picture. Now, knowing that the structure of DNA, your work on cell biology should identify, or you should be able to identify where proteins are made. And proteins are made within the cell at ribosomes. That's the site of protein synthesis. However, we've just learned that the codes for the proteins are in the DNA. Well, the DNA, as we know from the cell biology unit, is stored in the nucleus. Now, this DNA cannot leave the nucleus. It's like a very, very important library. We cannot have DNA floating around the cell. We've got 46 chromosomes in there, and we do not want them floating around willy-nilly, and we need them all organized so that we can grow and repair and etc. I like to use the analogy of here we've got a secret recipe. So the DNA contains a recipe for, let's say, making a cake that we want to make. The ribosome is where it's made, this cake. So we can call the ribosome the kitchen. That's where we make what it is we want to make. But we can't take the recipe to the kitchen because it's too valuable. We can't take it out of the library because it's too valuable. So what we have to do is we have to copy. We have to make a copy of the code that we want, or the recipe that we want to make, to take it to the kitchen. So essentially what we do is we make a copy of the recipe that we need, and we send it to the ribosome. That copy then lines up on the ribosome, and what we have there are little chefs that read the recipe, and they bring the right ingredients to the kitchen to make that special cake, or that protein in our case that we're making. So what's really important is that the ingredients for our cake are floating all around the cell. They're floating around the cytoplasm within the cell. So as this code is read, as the recipe is read, our little chefs grab hold of the right ingredient based on what what it codes for, and they bring those ingredients to the ribosome in the right order to make the protein that we want, or that the cell wants. Rather like a cake, the ingredients in a cake would be flour, eggs, milk, water, etc., sugar. Well, the ingredients to make a protein are amino acids. So these amino acids are floating around in the cytoplasm, and when they're needed to make a protein, these little these molecules, like these chefs, which is tRNA, comes along, grabs the amino acids, pulls it to the ribosome, and basically makes the cake. That's generally the big picture, and you need to keep that in mind when we go into all the terminology, the scientific terminology, and that's essentially the process. We make a copy, we send it to the ribosome, and then that's where we make the protein. So without further ado, let's go into more detail. So there are two main processes in protein synthesis. The first being what's written up here is transcription. Transcription is that first thing that I said on the last slide. Is basically it's a copying of the code. So in essence, what we do when we copy this code is we copy from a strand of dRNA onto a strand of mRNA, and the M stands for messenger, which is quite handy because really that's what it is. It's the messenger. It takes the copy from the nucleus to the ribosome. So here we've got a diagram showing transcription, and where are we in the cell? We're in the nucleus, and so here's the sort of nuclear membrane around it. Okay, what we've got up here at the top is a DNA strand, just exactly as I've described it earlier on. The sugar phosphate backbone in that double helix shape, and hopefully you can see that there are base pairs going one on one side, one on the other, and they're in those complementary base pairs, CG, AT. Chris Gill, awesome teacher. Okay, so what we've got here at this point is we've got the DNA splicing open to reveal the relevant code for the recipe that we want to copy. Now notice that it's only one side of the DNA, one strand of the DNA, i.e. this side, that's being copied. The other side, we can ignore it. It's not the one that we want. It's the code on this side of the DNA that we want. And so it exposes these base pairs. You know, you can imagine that the hydrogen bond's been broken down and the DNA splits up. And hopefully you can see these letters that start from, let's say, this point here. T, looks like G, G, A, C, T, T, G, G, A. Can't see that one, but G, C, T, T, G, C, A, C, A, G, C, T. Okay. And that's the original genetic code. But what you can see here in the nucleus, in the nucleoplasm floating around, are these free nucleotides. Free nucleotides. Remember what a nucleotide is. It's a building block of DNA, and it's made of sugar phosphate backbone and a base. And you can see that all these bases are different. C, G, A, C, G, U. Okay, and we'll come on to that one in a second. But they're ready and waiting. They're like, I don't know, like spare Lego bricks waiting to be used in the box, ready to build whatever it needs to build. So what we've got to do, remember, is make this copy of the code. And you can see that the copy has been made here. And this is our strand of mRNA. Our messenger RNA, and hopefully you can see that the base pairs, the nucleotides that have been made here, are complementary to the original strand of DNA, which is here. So as you move along, you can see that G matches with C, C to G, T to A. However, there's only one exception, and this is the main thing that sort of confuses students is that there is, and I'll write this in capitals, no T on RNA. And write a bubble around that. There is no thymine on RNA. It's replaced by another base called uracil. Okay, so remember, Chris Giel, awesome teacher, is no longer on an RNA strand. It's Chris Giel, awesome orangutan. So hopefully you can see right there on those base pairs, you can see that when A is on the core strand of DNA, the matching pair would not be thymine, it would be uracil. Students ask me why in the classroom. Really all we can say is that it's evolutionary and it's to stop problems in the genetic code copying over. We'll leave it at that. We're trying to learn protein synthesis, so you're just going to have to acknowledge that it does and move on. So what we've got here along this strand, I need another different colour. Let's do purple. This strand here is now our strand of mRNA. It's made of the same stuff. It's got a sugar phosphate backbone and it's got these bases that complement the strand of DNA, so it's copied. That single strand can then leave the nucleus through the nuclear pores and head on over to the ribosome, because that's the thing now that has the copy of our secret recipe that we want to make. So here's the second process in the two-part process of protein synthesis, and this is translation. And this occurs at the ribosomes. Now, a key link in knowledge, if you're thinking about cell biology, is that protein synthesis, cells can either make proteins for their own cell or they can make proteins to be exported around the body. So when you think back to your cell biology unit, those ribosomes that are on the rough endoplasmic reticulum are producing proteins that are exported because they're linked with the Golgi bodies and they package things in vesicles ready for exocytosis. Now, the ribosomes that we're going to look at here are the free ribosomes floating around the cell, and they're going to do things, they're going to make proteins for the cell itself. So what I should have said was that on the last slide is that here's the mRNA strand coming along. This is the copy, remember, it's just been copied. And what I should say is that every 3 nucleotides on this mRNA is called a codon. Okay, I should have said that last time. So when we looked at the structure of DNA, we said the structure was in triplets. That's on the DNA. Well, the corresponding triplet on an mRNA strand is called a codon. So now we've got a load of codons here on the mRNA strand with complementary nucleotides where T has been substituted for U. Okay, and that code, that recipe has been transferred to the ribosome, which is right here. It's this thing. So what we've now got is a special code that's arriving at the nucleus. And remember I said in the big picture, what we've got in the cytoplasm of the cell is a load of amino acids which make up a protein. And there are these circles here and here and here and here. These are the circles that are floating around. Now we only have about 20 amino acids in the human body that are used. And they're all sort of represented by these sort of 3 letters, phenylalanine, arginine, valine, lysine, I think it's tryptamine, I can't remember, but cysteine, whatever. But they are our ingredients to our cake. They are the building blocks of protein, amino acids, and they're all these purple circles here. Now what we've got are these sort of, I don't know, makes me think of playing marbles and the jack in marbles. But you've got these things here, which is the molecules of tRNA. That should be a little tRNA. So these are like our chefs. They're the workers. They're going to bring the ingredients together. So what happens is that we have a start codon, so that would say start, and then what's happening here is that the chef, which is the sort of cross thing here, the tRNA molecule, has on its feet the corresponding anticodon, which is, let me get a good colour to highlight that. This is the anticodon, which corresponds, I know this is heavy, to the codon on the RNA. Remember we said that this is the codon, so this is a codon on the copy strand. Excuse me. UUU. Okay. The chef has on its feet, I call it its feet, the corresponding anticodon. So where UUU should be TTT really, but there's no T on RNA. This would be TTT essentially. So the corresponding codon will be AAA, and that AAA corresponds to a particular amino acid. In this case, phenylalanine, which is this one here, I think. Yeah. So what happens is then this shifts along once it's dropped, once it starts. So this next tRNA molecule here has the corresponding anticodon to the next three letters or the next codon in the sequence. So CGA would be GCU. Remember, it's U because there's no T on RNA. So A, the A here would be T, but it's U because there's no T on RNA. That corresponds to tRNA and mRNA. They're both types of RNA, so it doesn't matter. So what happens is, as you can see on the arrows, that this next tRNA molecule, this next chef, comes along onto the ribosome, and what it does is it sort of cozies up the amino acid that it's bringing. Remember, it's the ingredients that make the protein. These two then will butt together. Obviously it'll be here because that's where it'll be coming, and these two will then join together to form a peptide bond, and we'll learn all about that when we talk about the structure of proteins and the primary structure, tertiary, etc, etc, etc. But that basically keeps on going along the next three. So we can see the next three and the next three and the next three and the next three. So it's like a conveyor belt of chefs just pulling these amino acids, these circles here, in the right order. So we end up creating this chain of amino acids, one after another, in exactly the right order that the DNA coded for in the first place. The result of that is that we get a specific shape of protein. So those people that have had the cafe lesson in our lessons where we're creating specific proteins, it depends on what order the base pairs were in the first place. So if we wanted a protein receptor, then we just put the amino acids in the right order and we get this shape, protein receptor for the cell membrane. If we wanted a protein channel, we'd have a different code and we'd end up having a protein channel on the cell membrane. And all that will be different between these two proteins is the order of amino acids, because these start folding up into the right shape to be whatever we need them to be. That corresponds to protein pumps, it corresponds to hormones, neurotransmitters, enzymes, anything to do with human physiology. When we're talking about proteins, we mean specific shape, and we've seen that so many times already in our sort of learning. So that's translation. It's basically getting hold of the copy of DNA and it's reading it and then bringing the right ingredients together to make the protein that the cell wanted in the first place. So now that we know the importance of this code, this DNA recipe, this code, we should now apply it to the things that you may well encounter in your profession in terms of health professions. Because if we change the code, if we change a single letter, then the triplet changes, and that may very well code for a different ingredient when the chef has to come to make the cake. So where, let's say, CAC might code for valine, if you change one of the letters to CAT, for example, when the chef reads it in the kitchen, he may well bring in phenylalanine instead, and therefore we'd have a different protein. And this is exactly what happens where in the condition of sickle cell anemia. So this is a nice little picture which kind of describes what's going on, but I would suggest that you, when you're writing your assignments, etc., you're looking into it and doing your own research. But normal red blood cells have 270 million hemoglobin molecules in their normal state, which means this one little red blood cell, this one little erythrocyte, has 270 million molecules of hemoglobin. And hemoglobin is a protein made at the ribosomes, and that function of that protein is to carry oxygen for us. So obviously without that ability to carry oxygen around the blood, we would seriously feel the effects of not being able to produce energy and whatnot. So sickle cell really is when the protein is made incorrectly, so we get cells that are, not surprisingly, a sickle shape, which changes the shape of the erythrocyte, which also makes it less efficient at carrying oxygen, and therefore has massive effects on our metabolism, and you can read that later on. But your task is really to explain how that happens, and if you've understood the previous videos, you should very well understand why sickle cell is caused, because here's the copy of the DNA. Now it's DNA. Okay, so you've always got to be really careful when you're looking at these diagrams to find out where these codes are. Is it DNA, is it mRNA, or is it tRNA? This is the original copy of DNA, and you can see that sickle cell is caused by a change in a base. The normal base would be T, but for some reason it's substituted for A. If you want to find that reason out, go and research it. But the key message here for this video is that we've changed a letter, right? Whereas the normal base would be T, we're changing it to an A. And if you want, you can look here on the sides, chromosome 11, that's where the base change occurs. That's where our code for this protein is. So if we change that letter, right, so where the DNA would be CTC, that's usually how it would be. Usually that's the triplet. That's the triplet on the DNA. The codon, hopefully you should be able to work that out. The codon on mRNA would be GAG. And when the chefs on the tRNA molecules come along, on their little feet they would have CUC. That's what would normally happen, and that would code for, let's say, phenylalanine. Let's have a look at what would happen if we change these letters, and hopefully it's quite obvious. The code would go from CTC to CAC on the triplet. The codon on mRNA would read GUG rather than GAG. And the chef, when he comes along, would read, well, you can work it out, CUC. A, C. So the chef's been told incorrect information. These tRNA molecules are being told the wrong triplet, the wrong codon, or wrong, actually anticodon. These are the anticodons on the tRNA molecules. So rather than be phenylalanine, this may be leucine. Now, you might not think that would have a massive impact, but changing that one letter changes the proteins, or changes the proteins from allowing a shell to be this shape to be a cell to that shape. And it basically, as the amino acids start folding up where they should fold up, let's say in this way, because the bonds between them just make it that way. That's what should happen. What actually happens in this case would be the fact they could fold up this way, and this is only an example. And so you got a completely different sort of shape in this area, and that, when you look at the protein as a whole, that's what causes it to come out of shape. And an out of shape protein is not a good one, because proteins have specific shapes, and that's what causes sickle cell anemia. Hope that helps. Okay, so I'm going to check your learning. The next task for you to do is to have a look at this diagram. I want you to try and identify A to I, which are the different landmarks and things that are involved in protein synthesis. And then I want you to go through the processes, so from 1 to 8. So make a list, 1 to 8, A to I. Pause the video, identify what's going on and what they are and what is happening. And then I'll go through the answers. So pause the video and the answers will follow in 3 seconds. So let's go through the landmarks first. We'll go through A to I. A is, you could have a variety of examples there. You could have DNA, you can have double helix, or you can have sugar phosphate backbone. It depends. It's quite ambiguous what it's pointing to. So give yourself a pat on the back. You've got the right answer. B are free nucleotides. These are the things floating around in the nucleus, made of sugar phosphate backbone and a base. C is a tricky one. It's an enzyme which creates the mRNA strand, so that's mRNA polymerase. D is what it's making, so that would be the strand of mRNA. E would be the nuclear membrane, and F would be nuclear pores, the holes where the mRNA goes out of. G would be the chefs in our process, so they would be our molecules of tRNA. H are the free amino acids that are floating around the cytoplasm, so they're our ingredients to make our protein, they're the building blocks of protein. I, easy one, would be the ribosome, and J would be the actual protein being made. So let's go through the processes from 1 to 8. Well, number 1, the DNA is splicing, it's opening up to expose the code that's needed. 2 would be the enzyme is helping to make the strand of mRNA. 3, the DNA would wind back together into its double helix shape. 4 is where the mRNA leaves the nucleus. 5, in fact I'm going to go out of order because the numbers don't match up. So I'm going to talk about, let's say 6 first of all, is where the codon meets the anticodon for the correct amino acid. And 5 would be when the tRNA molecule for the next codon brings the corresponding anticodon with the relevant amino acid into the next place on the ribosome. Excuse me. 8 would be the correct anticodon where the amino acid would join to the tRNA that corresponds to the correct anticodon. So that's basically when the amino acid joins to the tRNA. Have I missed anything out? And 7 would be when the tRNA molecule has dropped off its amino acid on the ribosome, it then goes into the cytoplasm to look for its corresponding amino acid. Job done.